Hello everyone. Today we are going to learn about the endocytosis and exocytosis. These are two important processes by which the materials move in and out of the cell. So what is endocytosis? First, let us understand endocytosis. Endocytosis means taking the substance into the cell. The process in which the substance is transported into the cell by unfolding of cellular membrane around the substance and internalizing it. It's simply engulfed or enclosed by these membranes, cell membranes. There are three types, and first, pinacocytosis, phagocytosis, receptor mediated endocytosis. Phagocytosis means it is a cellular eating. For example, white blood cells eating during eating the bacteria, whereas pinacocytes means cellular drinking. Here the cell takes in liquid, and receptor mediated endocytosis is very specific actually. Only the selected molecules enter the cell using receptors. So you can see through these pictures here the phagocytosis means it is a cellular eating. So the white blood cells eats the bacteria, where pinacocytosis means cellular drinking. Here the cell takes into the liquid. So third one is receptor mediated endocytosis, is very specific. Only the selected molecules enter the cell using the receptors. And then the reverse process of endocytosis is exocytosis. So materials are expelled or secreted from a cell is called exocytosis. So here the large molecules that are manufactured in the cell are released through the cell membrane. For really large molecules, vesicles forms, and it refuses to the plasma membranes and spills its contents out excretions to get rid of the secretions to push useful substance like hormones out of the cell. So examples of exocytosis are a release of hormones like insulins and release of neurotransmitters in nerve cells and so on. So let us go into the definitions. What is meant by exocytosis? Exocytosis is the process by which the cell releases substances from internal vesicle into the extracellular space, whereas endocytosis is the process by which takes in substance by external environment by engulfing them into the vesicle formed from the cell. So this is the examples. So what is the directions of exocytosis and endocytosis? It moves the substances out of the cells, whereas endocytosis move the substance into the cell. So I hope you all understand this. Let us go into the vesicle formations. So exocytosis and endocytosis. So exocytosis involves the fusions of vesicles containing substances with the cellular membrane, whereas endocytosis involves the formations of vesicles from the cell membrane to engulf the substances. And then fourth is purpose. So if we are seeing the purpose of endocytosis, it is used for the secretions of molecules such as hormones, neurotransmitters, and digestive enzymes, whereas endocytosis is used for the uptake of nutrition particles and signaling the molecules. So what are the energy requirements for the endocytosis and exocytosis? Generally requires energy in the form of ATP, that is adenosine triphosphate, to drive the vesicle fusion from the cell membrane, whereas endocytosis energy is required for the formations of the vesicles and the internalizations of the substances. There are types of endocytosis and exocytosis. It includes constitutive exocytosis, continuous secretions, that is, and regulated exocytosis that triggered by the specific signals, whereas it in endocytosis it includes the phagocytosis. It is an engulfment of a large particles. So pinacocytes, it uptake or fluids, and receptor mediated endocytosis. These will comes under the types, and let us see some examples of endocytosis and exocytosis. The endocytosis examples is it is an uptake of nutrition by the cells. It's lining the intestine, internalizations of LDL particles for cholesterol uptake, and phagocytosis of pathogens by immune cells, whereas in exocytosis it release of neurotransmitter at synapses, secretions of hormones by endocrine cells, releasings of digestive enzymes by pancreatic cells. These are the examples. And if we see about the membrane dynamics, for exocytosis, it involves the fusions of vesicles membranes with the plasma membranes. So it leads to the incorporations of vesicles contents into the extracellular space. Whereas in endocytosis, it involves invaginations of the plasma membranes to form the vesicles that encapsule extracellular membranes. So these are the membrane dynamics we have to see. So we have to see about the regulations. So by exocytosis, it is regulated by the intracellular signaling, the pathway, and the calcium ions. Whereas endocytosis is the different types of endocytosis are regulated by the various factors, including the receptors, cytoskeletal elements, and signaling molecules. So what is the role in cell communications? So if we see in exocytosis, it facilitates the intracellular communications by releasing the signaling molecules such as neurotransmitters and hormones. We have seen first itself. So in endocytosis, it allows cell to receive signals from the environment by internalizing the extracellular molecules. The receptors involvement in exocytosis and endocytosis generally does not involve receptors on the cell membrane, but in endocytosis there is a specific receptor mediated endocytosis. So it is specifically involves the receptors binding to the ligands for the internalizations. So what are the membranes will recycle in exocytosis and endocytosis? So it plays a crucial role in recycling the components of the plasma membrane, whereas in endocytosis it actively involved in the membrane by recycling the bringing into the materials for degradations or recycling. So if the transportation substance is taking in exocytosis, it is typically transport proteins, lipids, and other molecules synthesized by the cells, whereas in endocytosis it transports the variety of substances including nutrients, ions, and large molecules like proteins. So if we see about the speed of the exocytosis and endocytosis, if we see the speed of endocytosis, it is generally occurs at a slower pace compared to the exocytosis, whereas exocytosis occurs rapidly, especially in regulated exocytosis. So if we see about the cellular location, primarily it occurs at the regions of the cell membrane where the secretory vesicle dock. It's come under the exocytosis, whereas in endocytosis it can occur various regions of the cell membrane depending on the type of endocytotic process and specific molecules like being internalized. So by learning this, endocytosis brings the materials into the cell, and whereas exocytosis send materials out of the cell. So both processes are important and required energy. That's all. Thank you. Let us discuss about these questions and assessment. So what is the primary functions of endocytosis? Actually, it is an absorption of an extracellular materials. And what is the characteristic features of receptor-mediated endocytosis? It involves a specific interactions with the membrane receptors. So what type of endocytosis is characterized by the cell taking in a small solutes? It is a pinacocytes actually. If we has a quick comparisons, the endocytes materials enter the cells, whereas exocytes materials leaves the cells. So in endocytes, the membrane folds inward, whereas in exocytosis, vesicle fuses outward. So in endocytes, it needs an energy called ATP, and in exocytosis also, it needs an energy. In endocytosis, it uses the vesicles, where in exocytosis, it also uses the vesicles. So I hope you all understand everything. So thank you.